In this video, we will demonstrate novel techniques that allow us to image pupae of Drosophila melanogaster without compromising the structural integrity of the puparium. By keeping the pupal case intact, we minimize the impact of imaging on the development and viability of the pupa. We use a resonance scanning 8000 Hz confocal microscope for imaging. Importantly, the very fast scan speed and high sensitivity of this microscope greatly reduces bleaching, allowing for extended periods of observation. With this method, we've been able to visualize a variety of cell biological processes, including the behavior of phallopodia, morphogenesis, phagocytosis, and mitosis. With this technique, we can observe events in situ for extended periods of time without damaging the organism. The observations gained from these methods can be used to understand a variety of biological phenomena at this relatively inaccessible stage of Drosophila development.
To choose pupa at the appropriate stage, we examine them while still in the vial, looking for key indicators of developmental stage. To study cell divisions, we look for pupa that have everted their head but still don't show green packets of meconium in the dorsal abdomen. Individuals are loosened by touching the pupal case with a moist paintbrush, transferring water to dissolve the salivary glue. Pupa are then gently nudged free, picked up with the paintbrush, and transferred to a petri dish containing water. The pupa are thoroughly washed in the water to clean the case of food or debris. We then line them up, dorsal side up, to make sure they are of the correct stage. Handle them carefully. They can easily die if even a small hole is made in the puparium. Select individuals are carefully placed in the glass bottom petri dish and oriented so that the feature of interest is closest to the cover slip and as parallel to the surface as possible. To observe the pupal wing, we prop the pupa against a support, such as a thin length of modeling clay or dental wax, ensuring that they do not move after orientation. We then invert the dish to make sure that the feature of interest is visible and well oriented. Typically, mount multiple